Assalamualaikum and very good morning to all of you. Welcome to Malaysia Natural Product Society MNPS INBIOSIS Online Forum Series 2 2023. I'm Professor Dr. Nataqain Baharum, your moderator for today's session. I feel so grateful today that all of us are here, granted with good health, time, and have an opportunity to attend this online forum. This is the second online forum held this year by the MNPS, co-joined with INBIOSIS. I hope there will be more in webinars and online forum that will come, and hope all of you will join us again in the future. Here today, we have three distinguished panelists, Dr. Siti Zuraidah binti Mohamad Zobir from National Institute Biotech Malaysia, Dr. Darshan Singh from University Science Malaysia, and Associate Prof. Dr. Chyan Leong from INBIOSIS UKM. They are here to share with us their expertise, experience, and knowledge about our today's topic. I think this is a very hot topic in Malaysia, right? Ketum research in Malaysia, challenges and way forward. So without further ado, I would like to open the forum with our panelists today. Before that, I would like to introduce to you our panelists by reading their biography. So I tried to simplify the biography. Okay, so we start with Dr. Zuraidah. Okay, Dr. Siti Zuraidah binti Mohamad Zobir is a scientist and strategic planning and corporate development analyst at National Institute of Biotech Malaysia (NIBM) under the agency of MOSTI. At NIBM, her research works are in conducting Malaysia Institute of Pharmaceutical and Nutraceutical Malaysia (IPHAM), where she research interests include in silico, mode of action analysis, drug synergy modeling, system biology, and molecular modeling of natural product chemistry. Currently, she is a leading research project under the Strategic Research Fund MOSTI entitled Development of Depression Treatment for Metragana spiosa 2022-2024. She also has been as a co-investigator for two FRGS projects, which are a framework for the reconstruction of protein target activity prediction model of Malaysia therapeutic natural product using optimized deep learning approach and structure elucidation of bioactive compound. She also presented and published her research work relating towards understanding mode of action of traditional medicine by using in silico target prediction. Other than scientific research, she also a project coordinator for request for proposal impact study on National Biotech Policy 1.0. As administrative work, she was the head of unit National Product and Drug Discovery at the IPHAM and currently is the chair of NIBM Animal Ethics Committee and head of the QA NIBM IPHAM GMP plot plan for traditional medicine product. She also contribute on the Jawatankuasa Kajian Kebolehselaksanaan Pembangunan Industri Ketum under the Ministry of Agriculture and Food Security, and alternative member MOSTI Jawatankuasa Technical Traditional and Complementary Medicine Blueprint under MOH. So without further ado, I would like to invite Dr. Zuraidah for her first talk in this morning session. Dr. Zuraidah, you are welcome.
Thank you, Prof. Ain, for the kind introduction. Let me share my screen first. Okay, I hope everyone can see my screen here. Okay, a very good morning. Assalamualaikum and very good morning to everyone. Thank you for coming to this forum on the Ketum Research in Malaysia, Challenges and Way Forward. And first of all, I would like to thank you to the Malaysian Natural Product Society and MNPS for the invitation to be one of the panelists of this forum today. As introduced earlier, I'm Siti Zuraidah and actually a scientist at the IPHAM, or the long name is the Malaysian Institute of Pharmaceuticals and Nutraceuticals Malaysia. And at the same time, I am also being placed at the CEO office of NIBM as the Strategic Planning and Development Analyst. And at IPHAM, I work under the Natural Product and Drug Discovery Center. So let us go. So I'm going to give some briefing what NIBM is. Maybe a lot of people confused because they are the three institutes and there is a NIBM. So NIBM is actually a consortium of three different institutes being restructured and corporatized under the National Institute of Biotechnology Malaysia in 2014. So we have three different institutes at three different locations, and each institute has different focus area, such as IFARM. We are located in Penang, just across USM. We focus on the drug discovery from natural tropical sources for healthcare, pharmaceutical, and nutraceuticals. And the other two sister institutes of us are located in the Klang Valley. One is the Malaysian Genome and Vaccine Institute. This institute was transformed from Malaysian Genome Institute, MGVI, which was formerly known as MGVI. So in order to support the National Vaccine Development Roadmap, MVDR, the MGI has been transformed into MGVI to add the function of vaccine. So it focuses on the cutting edge research on genomes, omics, and human vaccine development. Last but not least, our third sister institute is the Agrobiotech Institute, which is located in Serdang. To be geographically correct, we are located in the Mardis compound. So the institute focus on discovery and development of value-added crops, livestock, food for better quality and products. So NIBM HQ itself are located in the complex of ABI, which is I am working at the moment. So just to have some like a small promotion that we also have accredited labs and facilities at NIBM, and all these accredited labs and facilities are located at IFARM. We have first the ISO 17025 for toxicology test. This is for the in vitro. And second, we have the facility, we call it as the good manufacturing practice, which we could produce the prototype of the traditional medicine product for the NPRE registration. And lastly is we have the animal research facility, which have been accredited with from two different bodies. One is the OECD and another one is ALEC that focuses on the preclinical toxicity testing for pharmaceuticals, cosmetic, veterinary drugs, food additive, and medical device. And ALEC is actually for the breeding program. So feel free to contact me afterwards if you're interested in one of in our facilities. Okay, so when we talk about Ketum research, actually at IFARM, we have started research in Ketum way back in 2007. That was during RMK9. And from the grants that we received, this is the list of publications that we able to produce from the studies, from the grants that we receive, from the grants. So if I could summarize here, most of the studies focuses on the mitragynine, the most active compound in the Ketum leaves. So in our publications, we found that the compounds has indication against the inflammatory conditions, and it could reduce the morphine withdrawal symptoms, both in vivo and also in vitro. So for the in vivo, we are using the model zebrafish. And then it also has shown cardiotoxicity effect and also some indication on the cytochrome CYP1A2. And the highlights of the Ketum research at IFARM, we managed to produce mitragynine with 98% purity. So the one from the method that we successfully developed here, the isolated mitragynine has been used to feed all the other research being conducted at IFARM at that time. Okay, when we look at the research of Ketum around the globe, this is the summary that we could find. This is actually data from 2021 from the Web of Science. In 2021, there are about 487 publications from all over the world. The most published countries is USA. Malaysia is one of the top as well. And there are also some European countries that also publish research on Ketum. Sometimes it's not about, the articles is not solely on research. Sometimes it's about the case studies or the abusive case studies of the Ketum extract, of the Ketum samples that they found in their country. Okay, so when we look at here, the worldwide publication, it has a growing interest. And if I'm not mistaken, the Nataqain was first reported in 1921. That is like more than 100 years ago. And the data that I get from the PubMed, it shows that the number of publications keeps increasing every year, which last year there were like 100 publications on Ketum. So the graph, the figure on the right side here is actually our very brief experiment or our brief analysis, because at this time we would like to see the similarity of the focus area of the research in Ketum across different countries. So this data actually originated from only about 159 articles in 2017 that we got from the Science Direct, and also the list of references from 2 review papers that was published in 2013 and 2016. So this figure, we call it as a heat map. As we can see here, at the lower, at the lower side, that where the similarities exist. So many of the countries focuses on the synthesis, analytical, pharmacology, pharmacokinetics, and etc. In total, we found that only 20 different focus areas from these 159 articles. Maybe if we would like to see how the trends look like right now, we could actually get new data, number of the, articles until 2022, and see what is the trend all across the globe, the focus area of research Ketum being conducted. Okay, so from there, we know that because there a lot of data has been published, so there's a lot of data generated as well. So in many of the findings, we know that there are secondary metabolites in the Ketum leaves, and it's not only alkaloid. They also found the presence of other phytochemicals such as flavonoids, saponins, tannins, phenol, and steroid or terpenoids. So on alkaloids, there are about more than 50 compounds being identified. And from the pie chart on the right here, it shows that metraganin, the percentage of metraganin is the highest among all other alkaloids, followed by the other compounds stated here. If we see, actually most of the study focus on the pure metraganin, and on the other alkaloids need more exploration. And we know that metraganin also has a very poor solubility in water and it's very unstable. And from the studies conducted scientifically, it shows that metraganin could reduce, has the bioactivity as pain reliever, addiction recovery, energy booster, reduce anxiety, immune system booster, diabetes treatment, and antidepressant. So what we came about at IFARM in 2021, this made us thinking. If metraganin has a very poor water solubility, and a lot of negative perceptions, especially in our country, the negative effects of taking the Ketum leaves. But this Ketum is likely used by 3 to 5 million in the US for diverse health reasons. We couldn't find any data for Malaysia in Malaysia, so that's why we are using the data from other country. And Ketum positive death accounted less than 1% for overdose deaths during July 2016 and 2017. So we were asking ourselves whether metraganin itself is actually the only compound that has this the most the most contribution in the activity of the Ketum leaves, or is actually the set of compounds in the Ketum leaf itself. And we are looking at the depression. This is based on the study reported in 2011 by Idayu et al., finding that mitragynine exerts antidepressant effect in animal behavior and mode depression. And secondly, why we picked to go into depression, because we wanted to complement research that have been conducted in Malaysia. Like USM, they are really expert in the pain management. So we would like to see at a different angle of the use of Ketum leaves, so that we could actually find something tangible from our own native plant. Okay, this is the current research that we conducted. It is a 3-year research, and the fund was from MOSTI. It is called the Strategic Research Fund. So in this case, because like I mentioned before, we are looking at the set of compounds. So we are looking at the standardized extract of Ketum instead of the mitragynine itself. So how we come across this, this is based on a lot of publications on the traditional Chinese medicine, because in TCM, they have a different formula, and each formula is a set of herbs. It's not only contain one herb. So researchers in China, they have studied very extensively in order to decipher how the mode of actions of all these compounds in the one formula in order to tell, or in order to explain how this formula works for the indication that have been put in the old manuscript. So in our case, the Ketum standardized extract will consist a lot of compounds in there. And then we will do the chemical profiling in order to get this set of compounds, and we also will do the physicochemical characteristic. From there, from the set of compounds that we gain from the first process, or from the first phase, we move on to the molecular chemistry, which we will do here. At this phase, we will do some in silico target prediction and pathway screenings, and we will develop a network biology and to see where all these targets of the compounds in the network biology, and we hope to decipher one of the molecular interactions from the most potential target. And we also will move into the in vitro. This is to test the cytotoxicity and drug interaction of the standardized extract. And lastly, we will move to the clinical for the efficacy, safety and efficacy study. We're also going to look at the genotoxicity, pharmacokinetics, pharmacodynamics, and metabolomics. So this research, our collaborators are our neighbors, USM, and we have IMR and also FRIM on board to conduct this study. Okay, but as we know, Ketum has its own challenge because it is listed, the compound mitragynine itself is listed in the third schedule of the Poisons Act 1952. So we have a bit of challenge when we would like to conduct this study. And when we look at the poisons regulation in 1989, a person who handled this sample need to be usually a registered pharmacist, which unfortunately IFAM or NIBM has no registered pharmacist. So in this case, we need to get the special approval from the Director General of Health before we could start the study. So it's not a simple application, and we thought that probably like 6 months will be enough when we put the proposal in full, and we said that maybe the first 6 months we will go for the MOH approval from the Bahagian Penguatkuasaan Pharmacy. And once we submitted our application, they have asked us this information, the one on the left here, and we have to provide all the information before they could process and arrange for the lab or premise inspection. So this part has already taken about 2 to 3 months before the lab inspection being conducted. I think we received we put in the application end of 2021, and the lab lab inspections will help in I think in April or May. So it took a long time for that. So in the lab inspection, there's a lot of things that we did not know upfront, such as like all the Ketum samples have to be in the isolated storage with lock, and then we should prepare record in and out of sample from one lab to another. We need to declare which labs we are using. We just cannot simply set the whole IFAM. We have to specify which labs we are using. And then for the fridge, where we'll store the metragynine compounds, need to be locked. Need to have a temperature reader, and then there must be a storage key for traceability record. So there's a lot of things, and we also need to prepare like two different SOP, one on the safety and one on the disposal. So for the safety, we need to know like who actually hold the keys, and who actually can access to the labs. So they need all this information. And at disposal, they have told our friend that at the end of the study, when there is a extra or remaining of the sample, either metragynine or the Ketum leaves of the Ketum extracts or whatever, the disposal has to be done in front of their officers. So it has to be witnessed by their officers. And because of this, it incurs some costs. It also incur times and energy from us, because we need to respond to the compliance or readiness of the lab, especially on the fridge and also the storage, because as we know, like all our labs, we just put everything together. We never separate unless it is really like in this case. So we have to buy like new 2 to 3 fridge, because for this study, we will be doing the study one in MGVI, one in IMR, and also frame. So there's a lot of things need to be done upfront. And we only get the approval by the Bahagian Penguatkuasaan Pharmacy in August. So it took us 8 months in order to get this approval. So that is the challenge, at least for NIBM, in order to run the study on Ketum. And secondly, and because this approval is per project, we cannot simply conduct another project that will use the same sample in order to get a different output. Because the officer will come and do the random inspection every year to see whether we comply to the requirement that they have set up front. Okay, so I'm sorry, I couldn't move the slide.
Okay now?
Okay, tak boleh move slide dia.
Boleh share, unshare semula? Unshare dan juga share
how-
semula. Okay.
Okay, sorry. Okay. Okay. So the next challenge that we found when we do this study, when we started this study, is the source, is the way to get the leaves. So we are actually new, as we have to start back everything on the Ketum research after being paused for like five years. So thanks to Dr. Darshan, actually, he was the first one that we met in order to get advice where to source the Ketum leaves. So he brought us to the Bukit Bertajam Penang to see the farm, and also we also look for another potential farms from the Jabatan Pertanian. So we found we went to the Padang Terap Kedah and also Arau Perlis. Okay, the challenge here is that the location of these farms. So it depends on the soil types, whether it's rural or developed areas, and it will affect the heavy metal analysis and microbial contamination test, which we saw in our analysis. And then the size of the farm as well to support the production, because in our case, we would like to develop the standardized extract, water aqueous extract of Ketum leaves. And we declared to the KKM that we will source up to 200 kilos of fresh leaves in order to support the study. So the size farm might not be supporting for the production of 1 kilo standardized extract in our case, because we would like to, after we optimize the standardized extract at the lab scale, we will move it to the industrial scale, which one of our collaborator, which is FRIM, will be doing it. So in this case, we feel that the Ketum leaf extract should be standardized for alkali content, microbial contamination, pesticide, heavy metal, residual solvents, and along with other proper labeling requirement. So this is part of the requirement that is needed when we want to move our output to a product. Okay, this is the way forward. So when we draft up the proposal, we did not only stop at the preclinical stage. We also came up with some ideas saying that to go for the drug will take a long time. So maybe we could look at the nutraceutical product, or we call it the traditional medicine product. So this is what we anticipate to do in the next phase if the first phase successfully completed. But again, we have to remember that it's always a limitation, because according to the drug registration guidance document, the RDG, the latest edition, because metragynine is listed under the Schedule of Poisons Act 1952, so there should be no product coming out from this plant. So we hope by the time we are moving to phase 2, the study being conducted at MAFS, Ministry of Agriculture and Food Security, on the feasibility study of Ketum will end, and some good outcome from the study will be shared to all of us, the researchers. Okay, so and the second one, when I talk about the no standard farming, I think the way forward is also to have the good agriculture practice. This is to reduce the risk of harmful contamination of the plant, because in a GAP, information is collected systematically based on the plant morphology, production costs, agriculture practices, harvest and post-harvest studies. So this will increase the farm productivity and also could create income from small farmers engaged in the Ketum farming in Malaysia. Because when we went to all these farms, they have the similar concern being shared, that is like, if these plants could be legalized, so that they could sell it to at least to the researchers, so they could have some extra income to the family. And then we need more support from the government. Maybe government could allow Ketum cultivation on large scale under the regulations of any of the authorities or department under the respective ministry, to ensure a large and continuous supply of Ketum beans to local and international research institute. And the government to taking swift steps to patent the benefits of Ketum, anything that we found from our study, from our own researchers, to conserve our native plant. So this is what we could think of the way forward for this research. Okay, so I will end this. I would like to give this acknowledgement to our CEO, Prof. Datuk Wiraha, and our acting director of IFARM. And also these are the list of science researchers who will be part of this study that will end in 2024. And if you have any things to talk to us or any discuss to us, you can also contact two of my core team members, that is Encik Hadi and also Cik Azimah. With that, I thank you.
Okay, thank you, Dr. Zuraidah, for keeping up to the time. Okay, I think we move forward to another panelist for today forum. So I would like to introduce Dr. Darshan Singh. Okay, Dr. Darshan Singh is a senior lecturer at the Center for Drug Research, University Science Malaysia, USM. His key research focus includes study the emergence and abuse of the psychotropic substances in the, identifying addiction, treatment, and harm reduction prevention challenges among people who use the drugs. In addition, he has a strong passion for Ketum research and has conducted many studies to determine Ketum traditional use as well as health risks and effect among the people that are using the drugs. Moreover, he also investigates treatment barriers among the opiate users, HIV risk behaviors of the amphetamine type stimulants user, treatment barriers and need for of female for using the drugs. Dr. Darshan also researched on the strengths and weaknesses of the drug policies like the death penalty and corporate punishment. He worked closely with out of treatment and the people who using the new psychoactive substances. Dr. Darshan also actively participates in the university community-based engagement program targeting the communities plagued with the drug abuse issues. Indeed, his research aimed to help improve drug policies and laws for the betterment of the people among people who use the drug PWUD and to meet the UN Sustainable Development Goals. Okay, so I would like to pass the floor to Dr. Darshan.
Thank you very much, Dr. Syarul, for the lengthy introduction about me. Without further ado, I will begin to share my slides now. All right, can you see my slides now? Dr. Syarul, can you see my slides?
Yes.
All right, okay. So salam sejahtera and good morning, and thank you for inviting me to share my thoughts on a very important and controversial topic, like what Dr. Siti did mention, which has been blown out of proportion with nebulous information, making people believe that Ketum is another class or type of narcotics. Therefore, its planting and consumption must be strictly regulated as other illicit psychoactive substances in the country. Now, hence, this webinar series, I believe, may somehow help to allay or debunk the disinformation or misconception held by most people about the almost extinct Ketum using tradition in Malaysia. Let's move into the challenges. There are several challenges, and I will take you into each of these challenges. Let's look into challenges one. Now, I will now begin to share my 2 cents worth of thoughts on some of the pertinent challenges Ketum researchers, especially social scientists like me, encounter while conducting Ketum research in the country. Now, among the challenges, social scientists are finding it rather quite difficult to approach and get Ketum users to participate in field-based studies. Literally, Ketum users assume that their participation in field-based studies can cause Ketum to be outrightly criminalized. In fact, there have been situations where researchers have been castigated, people like me have been castigated, prevented from accessing Ketum pots in the community, and requested to comprehensively explain to these Ketum users about my study aims. You know what? I really envy those working in the labs, as they do not need to experience such rowdiness or bear the brunt of fieldwork hazards. Many times, as a social scientist working with people who use Ketum in the community, I have been conducting my fieldwork when Ketum pots come alive during late evenings, because if you were to access Ketum pots in the daytime, nobody is going to be there. So as a social scientist, like I was telling you just now, I really envy people working in the lab, because you do not need to experience all kind of public shenanigans like me, right? Okay, let's move on now to the second point. Now, sadly, I also hear people, including relevant authorities, concocting a negative narration on Ketum. Even the media have injudiciously reported on the bad side of Ketum and failed to educate or inform the public about the advantages of Ketum, right? So apparently the news depictions have made people to assume that Ketum is detrimental as illegal drugs, right? So for me as a social scientist, I've not seen people using Ketum developing significant social impairments, right? So when researchers go out, researchers like me, when we go out in the community, we face mounting pressure. Ketum users keep asking us so many questions. Always bila kita masuk ke dalam, always when we go into the community to work with them, they keep asking, tak habis lagi ke? You have not completed your studies. You're coming back again and again and again and again, and you have completely failed to legalize Ketum. At times, I have no proper answers for their queries. I see many people, take advantage to drum up an issue. They use adolescence as an Achilles heel to convince the government to take immediate action. This is what has been happening with the media depictions. They always target these poor adolescents not knowing why are adolescents involved in Ketum use. It's very simple. And you know that people who are dependent on illicit drugs are actually using Ketum as a substitute. And of course, these adolescents who are involved in drug use are actually using Ketum to self-treat their substance use disorder. Let's move into the third challenge. Now, since Ketum's most abundant alkaloid is being regulated, it is legally wrong to work with Ketum if researchers fail to obtain approval from relevant authorities, like Dr. Siti did mention earlier. It is very, very difficult to obtain approval. You have to meet certain requirements. Okay, so now, like she was saying, at times it can be very nuanced to obtain an approval given the rigmarole process. Because, like Dr. Siti was mentioning again, if you were to work with Ketum or if you were to work with mitragynine especially, right? You need to make sure that you comply with necessary procedures. Otherwise not, you are going to experience a lot of problem. So I urge researchers, if you want to buy back fresh or processed Ketum juice, because like me, I normally buy back fresh Ketum leaves or processed juice because I want to determine mitragynine content, for example. You have to safeguard yourself because the current laws allow authority to detain you until proven guilty. So don't simply go out in the community saying that, I'm a Ketum researcher and I want to buy Ketum. And on the way back, you could be intercepted by the police. And if you have no prior approval from relevant authorities, you are going to be in hot soup. All right, so remember. Now let's move on into the next challenge. Now, what we get to see is, unfortunately, those studies have shown that Ketum is being used for its vast beneficial effects, especially among people struggling to refrain from drugs. Now, against this background, I personally believe people caught for using Ketum should not be detained or jailed. Those who have been penalized by the criminal justice system are very reluctant to participate in field-based studies, and consequently, this will undermine prospective studies. Like me as a researcher, people are so frustrated with the criminal justice system because they are being caught for small Ketum related offenses. So when I go in again to conduct another study in the community, they always assume and believe that I am a police informer coming and telling to the police what is actually happening in a particular area. So because of this issue, they are so reluctant to participate in my, in my ongoing projects. And I have to sometimes plead with them and explain to them that I'm here to do something to keep Ketum legal, but they are saying how long? It has been almost many years. It has been 20 years since Ketum was first regulated under the Poisons Act in 2003, and it has been 2023. You can't even make a decisive decisions to decide whether Ketum should be criminalized, decriminalized, or what next. All right. Okay, let's move on. Right. So like Dr. Siti did mention just now that they have visited several Ketum plantations to profile Ketum plants. Now again, the most crucial point which I would like to highlight here is while some studies, I'm not saying no studies have been done, some studies have been done to profile the Ketum plant, whatsoever, the widely available Ketum plants in the country have not been scientifically studied. Remember, the widely available Ketum plantations or widely available Ketum trees in so many plantations across the northern region state has not been scientifically characterized. All right. Now, due to the high Ketum demand, planters have now altered, they have altered the original Ketum trees once found near the river banks, and now they are planting them on commercial plots, right? Now, given this limitation, researchers face problems working on a standardized material. Now, everyone, like Dr. Siti is saying, even me in my center, we are blindly buying Ketum leaf from different sources, and almost all of us have no idea about the quality of this leaf because there's significant variations in mitragynine content, during different seasons, right? So it is very important if we were to move forward into clinical trial, we have to make sure that we have a standardized raw material to support our future Ketum research. All right, so it's very important that more emphasis is being done to actually study about these Ketum trees in the country. Now let's move into the next what they call that next challenge. Now, in the last few years, as a social scientist, I have observed Ketum research has been so asymmetrical, it is so skewed, and a lot of emphasis has been given to only studying mitragynine, though people in the community have been consuming Ketum decoction. And if you were to see, from the very onset, maybe about 15 years ago or maybe 16, 17 years ago, since Ketum was regulated in the country, we get to see a lot of studies were looking at the abuse potential and the side effects of mitragynine. What we were doing was completely wrong because people were not using mitragynine per se. They were actually using Ketum decoction. All right. So it's very disturbing to learn that the data from rodent experiments have been used to make all sorts of inferences on Ketum, such as it has an abuse potential as morphine, and its regular consumption can cause adverse effects. I think so we have to stop all this kind of speculation. All right. Now let's move on to the next challenge. Now, while we have a serious and expanding heroin abuse problem in the country and persist to punish people who use drugs, instead of treating, sadly again, we see researchers giving very little importance to understanding Ketum's applicability among heroin and methamphetamine users. It is really, really shocking to me because when I first started on Ketum research years ago, I get to see a lot of heroin users were actually using Ketum. And after some time, I get to see people with polydrug use problems, heroin and, stimulant use disorder, methamphetamine use disorder, are also using Ketum. And lately in my current studies, I get to see a significant percentage of people who are dependent and struggling to abstain from methamphetamine use are actually using Ketum. I think so the research should now give more importance or equal importance to methamphetamine use disorder, because around the globe, currently there is no medicine-assisted treatment for ATS use disorder. So I have not seen any studies being conducted clinically to attest Ketum's utility for opioid or methamphetamine use disorder treatment. Even with these live experiences of heroin users, nobody wants to take this as a gospel truth to advance their research in finding a new antidote for heroin addiction. You already know clearly hundreds and thousands of people in Malaysia are using Ketum. Heroin users are actually using. You're not looking at how do we develop Ketum or mitragynine or whatever it is. How do we turn this into a medication to treat heroin use disorder? We are doing things, we're doing all kind of weird things. All right. Okay. Let's move on to the next challenge. Now, in addition, Ketum research is not streamlined. I'm telling you, as a social scientist, in the last 20 years, since Ketum was regulated in the country, we get to see people doing all kind of research in the country. So it's not streamlined. Everyone is trying to do something, I think, to meet KPI targets. I guess after today, I hope those who are involved in Ketum research will give more importance to studying Ketum's analgesic properties. You already know what mitragynine has better efficacy than morphine in treating pain. So why are we not looking into the analgesic properties of Ketum? And we're doing all kind of different things. So it's very important that we give more importance to studying Ketum's analgesic properties, or determine whether can Ketum be developed as a novel treatment for opioid use disorder. I see people busy speculating and reporting unfounded narrations about Ketum's lethal effects. We are so stuck with mitragynine, harmful, mitragynine at higher concentration is harmful. Again, as I said, people in the community do not ingest mitragynine alone. They use Ketum decoction. Let's move on into the next challenge. Now, last but not least, it is equally important for clinicians to develop treatment guideline to treat Ketum dependence and withdrawal in people presenting with people presenting with severe Ketum dependence. The guideline, I believe it's urgently needed to stem treatment providers from simply using their own discretions to treat Ketum users with the highly addictive methadone therapy, right? So I'm afraid in years to come, we will have to deal with a new problem, Ketum users frequenting methadone clinics. Now, I've seen so many GPs now, when I talk to them and I ask them, are you treating heroin users? Because we as a researcher, social scientists, we want to know what is actually happening on the ground, a lot of heroin seizure, what is happening to these people. When we go out, when we talk to GPs, we get to hear that they are busy treating people with Ketum dependence. So the guideline, it's very, very essential, all right? Okay, now I will take you into the next part, the way forward, which is very important, I believe. Now now the big question is, how should we unfold or aggressively progress forward with our Ketum research? Should we run, crawl, or sit and wait for durians from Balik Pulau to fall and then decide what to do next? I think we have reached a situation where we can make more informed decisions. Now is the time for us to move forward into translational research, enough of preliminary studies. What is urgently required now is clinical trials. So using whatever we have discovered over the years and charting new research trajectories. So pharmacologists, if you want to do good science, I think you have to work with social scientists. If all of us want to collectively do good science. Now let's look into the first way forward. So I believe in order to make a profound impact, more attention must be given to developing the nascent ketum industry or ketum crop as a valuable commodity in the country. This is a sensitive statement which can be interpreted wrongly. I'm not trying to promote the ketum industry, but I'm just trying to suggest. However, based on my personal views, which I express with some discretion, commercializing ketum doesn't mean that we will simply allow people to use ketum as they like. All right. But from an economic perspective, ketum cultivation has the potential to replace non-profitable commodities in the country. So I think so this is very important. Even the Kedah Menteri Besar, even our former health minister, Khairi Jamaluddin, did mention about we move towards exporting ketum leaves. Again, like I was telling you, many people were very unhappy with that decision. But again, like I was telling you, we have to really scrutinize. We have to really look into the pros and cons of what we plan to do, not simply go into exporting ketum. Why not, instead of exporting, why not we become the producer? We sell the end product and we generate more income, right? Okay. So as a social scientist, I always believe, and I did mention, our research is not streamlined. Everyone is competing, all right? So most importantly, after further analyzing the current context, I personally believe that there is a need for establishing a dedicated ketum research institute. For example, take the palm oil industry. When we first promote palm oil, nobody wants to buy Malaysian palm oil. Now we have become the world leaders in palm oil research and the largest exporter. How? Because we have dedicated palm oil research institute, which has never pulled its handbrakes on research until today. So the research institute, I personally believe, can play a bigger role and provide grants to aspiring researchers. There's so many ketum researchers. This is a very healthy development. But again, the ketum research in the country has to be governed by a ketum research institute. Let's move on into the next challenge. There are many alkaloids in ketum. Some minor alkaloids are shown to have some medicinal value. However, these alkaloids are not properly studied. In fact, researchers must give more importance to studying ketum decoction and the combination of other alkaloids in controlled clinical studies. We must leverage on the life experiences of heroin users and develop new antidotes for heroin dependence. So you already know you can use mitragynine to treat opioid use disorder. But again, like I'm telling you, in the community, if you were to look at the local studies, people are using ketum decoction. So I think so we have to move towards that direction seriously and coming up with a proper formula and moving towards clinical trial. Now, if we plan to export ketum, then we need to amend the current laws. It's very important to amend the current laws. We don't want to simply encourage people to go into ketum planting business. There's so many ketum plantations now. Everybody wants to become a businessman or grow ketum as they like and then sell it illegally. Yeah? So if the government is serious in exporting ketum, then we really need to produce a ketum planting guideline. Like Siti was mentioning, you need to have a proper planting guideline, to facilitate Ketum planters to follow the necessary steps to ensure their yields are of optimum quality. We are not going to simply go and buy, because now if I were to ask you, when is the best time to harvest Ketum? Now, what we are buying, they are saying it is maturely, which is about 4 weeks old, which is about 5 weeks old, which is about 6 weeks old. Do we have a definite time period to show that the mitragynine yield or whatever it is has reached its optimum level, and this is the best time to harvest Ketum leaf? Tak ada. There's nothing, nobody knows anything, and here we are busy doing all kind of research. Right, let's move on. So we have been arguing since 2012 in Parliament to harshly criminalize Ketum. The war on drugs ideology mooted, copied and pasted in Malaysia have failed in every dimensions to address the worsening drug abuse crisis. Now, we see hundreds of thousands of people who use drugs are using Ketum as a substitute to self-treat their substance use disorder. So we can decriminalize Ketum, but we need to inform and implement some laws to preclude people from misusing or trading it openly in the community. We can learn from the we can learn from the Thai, but again, we can draw our own laws to befit our current context. So you don't need to copy exactly what Thailand did. We can, we can give and take, we can tighten our intervention. So because we don't want people to start selling Ketum juice in pasar malam all if we were to decriminalize, okay? Right. So hopefully no one throws any brickbats on me as I delve further into promoting the Ketum industry in Malaysia. I think we are at a crossroad now, still deliberating cautiously whether to export Ketum or not. Now, how to export Ketum when everyone is saying Ketum is harmful and pressing the government to impose a total ban on Ketum? I think we have had enough and should just discard our negative, whatever negative information or thoughts that we have on Ketum, which continue to mentally disturb us. We should look at the bright side of Ketum. Ketum is $1 million industry now, and we don't want to lose out from this huge investment. Now, last but not least, I pessimistically believe that we are in the leading position. We are in the leading position to revolutionize the Ketum industry and research in the country with our own uniqueness. We have so much of uniqueness, all right? I don't think so anybody can compete with us when it comes to Ketum research. We can be the pioneer in developing novel treatment for pain and opioid use disorder. Remember, research has to be geared towards pain and opioid use disorder. And in doing so, we need to get the right support, do the right thing, and bravely believe in our research. Most of the time when we do research, end of the day, we don't believe in our findings. This is an irony. So it's time that we move from the bench side to the bedside, not the backside. Remember, from the bench side to the bedside. In other words, how we use our preliminary findings to move into clinical trials to benefit people struggling to cope with pain and opioid use disorder. And before I conclude, I would like to thank everyone for participating in this webinar and listening attentively to my presentation. Good day, and now I pass the floor to the moderator, Dr. Syarul. Thank you so much.
Thank you so much, Dr. Darshan, for the very insightful about Ketum in Malaysia and also on the social aspect. So I think we have a lot of questions now. So, but before that, we will continue to the last final panelist for today's session. Our researcher at INBIOSIS. Okay, I would like to introduce Associate Professor Dr. Chyan Leong. Okay, Dr. Chyan Leong graduated from UPM with a Bachelor Science in Biotechnology and Master Science in Genetic Engineering and Molecular Biology. He completed his PhD in York Structural Biology Laboratory at UC of York. in 2007. He then worked as a postdoctoral fellow at the Medical Research Council, Laboratory of Molecular Biology, Cambridge, UK, from 2008 to 2012. Dr. Ng currently is a senior research fellow at INBIOSIS and serves as head of the Center for the Genome Analysis and Technology Center and Plant Biotechnology Center at INBIOSIS. Dr. Ng research focuses on the 3D structure and function of micromolecules, including conserved hypothetical protein of human and pathogenetic bacteria, secondary metabolites, biosynthesis enzyme, and toxin and epitope of allergen molecules. Recently, his research interest has been extended to understand the impact of carbon sources in fungi metabolite biosynthesis and also study the alkaloid biosynthesis pathway in Ketum using the system biology approach. The research output of Dr. Ng Research Group and collaborator have been published in various peer-reviewed index journals, including Nature Communication, Scientific Reports, Plant Physiology and Biochemistry, Phytochemistry, PLOS ONE, Microbial Cell Factories, and H. His group recently reported the alkaloid repertoire in young and matured leaf of Ketum through comparative metabolomics analysis. So without further ado, I would like to invite Dr. Ng Chang Leong. The floor is yours.
Morning, sorry. Let me share my slide. So a very good morning to everyone. Thank you, Prof. For the introduction. Thank you, Malaysian Natural Products Society and Institute of Systems Biology for the invitation as one of the panelists here today in this morning to discuss about the Ketum. I start to have an interest to work with Ketum since around 2015 or 2016. And also thanks to a Center of Drug Research in USM, Prof. Sarif also at the beginning to invite me to the symposium in the Ketum in USM. And from there on, we are interested to discover the Ketum using system biology and also omics studies. So bear in mind, later the slide may be a bit in detail, but I would like to share in different angles on what we know about Ketum and also relevant to what Dr. Darshan and also Dr. Siti Zuraidah have shared just now. So it's brief introduction, and we in overview or overall, I will share about the current updates in the genome and metabolome study that related to Ketum, and the pathway, that how much we know about how this plant producing the active compound, the mitragynine, and also other alkaloids. And finally, we'll share a bit of our latest finding in comparing the leaf and a young leaf and also the mature leaf of the Ketum. What is the difference between the alkaloids compound or secondary metabolized compound in it? So Ketum is belong to this Lupicea angiosperms. In this family, they have 650 genera and contain 13,000 species. And genus Mitragyna actually having 10 or 11 species found in Asia and Africa, as listed over here, about 6 species in Asia and about 5 species in Africa. And apart from the pharmacology properties of the mitragynine or 7-OH mitragynine that has been widely studied and researched, there are also other properties of the extract or the compound found from other species. apart from Michoacana spiosa, and other species also reported to have a pharmacology properties. And I went to IUCN list, and most of the species there are 5 species over there. Michoacana speciosa is rather stable, as we know, everyone interested in it, so it's very stable. But there are species that are in the near treated, and also in the least concern, in a way that is decreasing the number of plant. So over here, we are interested in how actually the plant producing this compound, how much we know about it, and from the genomes, gene expressions point of view, and also what kind of metabolites that producing, like just now Dr. Darshan had mentioned, standardizations, right, from different geographical area or different season harvests, how much that we know from the molecular level for this. And it is important to understand this detail for understanding the alkaloid biosynthesis pathway and also the production of the bioactive alkaloids. So this is several questions that we ask and always interested in it. And apart from what we are doing on comparing the young leaf and also the mature leaf, there are area or that is important or fundamental. For example, how much we know about the genome of the Ketum, right? And how the gene expression pathway actually influencing the production of the alkaloids. And so even though we didn't conduct the genome research, but recently there are several studies have reported the genome and transcriptomes, and also intensive study on different cultivar and also different organ from the different tissue from the plants. And over here, I will share what we have learned or from others research reported recently. And for example, the first genome or draft genome was reported 2021 by the group from the Michigan State University. And the cultivar or variety that were used is a Michoacana spiosa reflux, and the draft genome size about 968 megabase. While another genome a year later was reported for Ketum from Thailand, and was using different technology, like biotechnology. And the size of the genome is about 772 megabase pair, and with a 22 chromosome, and it was found as a diploid chromosome over here for the genomic data, sequence data. So in first study, it predicted about 55,000 genes, and in second study, about 43,000 genes. So what we know from both studies also, they mentioned that likely the plant is a tetraploid, also based on the literature before, and also based on the Darby chromosome staining from the study that just now I mentioned, one of the studies over there. And from analyzing the genome, it found that the plant is very similar to the not say very similar, but similar to a coffee species compared to others. The distance or they divided 47 million years ago. And while in compare with tomato, it was hundreds million years or 106 million years ago from the last ancestor. So of course, with the data from the genome, we able to, from the genes that are annotated, we are able to know what genes is actually encoded for what kind of function. And you can see over here, the alkaloids metabolic process genes also found, and many genes or a number of genes are identified which may involving in synthesizing the active compound nataqain. So the study from the genome research of genome sequencing research for this Thai Ketum from Thailand. So they also did or collected the 85 species of the Ketum and study the differences, or rather look at the single nucleotide polymorphism SMP as a marker to study how similar they are. And they group the 85 species to two subpopulations and suggest that it could be a geographically associated, and in this case, Western and Southern in one population and Central Thailand in one population. I think this is relevant to what just now Dr. Darshan had mentioned. How do we know, how much we know about the Ketum species that in Malaysia or from different stage. So I think we don't have yet this data of to compare different variety or cultivar of the Ketum in the country. So I think that is one of the things that we need to pay attention. I agree with Dr. Darshan on that sense. So we also ask question that's different trees cultivars producing different amount of metabolites. So apart from they look different, are they producing different metabolites? So another study showed that by comparing different cultivar, so over here in this study done by Satya, Svati, Nakakuduti groups, and they reported that from the 7 cultivar, they indeed, this is a different morphology of the leaf, they're indeed producing different amount of active metabolites, in this case, mitragynine. And these 10 alkaloids or indolkaloids are common metabolites or alkaloids that we found in Ketum. And we can see that from different cultivar, the production is different of each of the metabolites. And 2 of them here, just would like to highlight is so-called Malaysian G, and in the paper, and also number 5 cultivar is Malaysian somewhere here. So if you can see 1 and 5 does have a clear difference in, if we look at the mitragynine content. So 1 would be a mitragynine dominant, and the 5 would be mitracyanine dominant. So when they did the DNA barcoding, use ITS 1 and 2, they also found that different cultivar over here that in the study show that they are close to each other, correlate to the production of the mitragynine in this case. So this suggests that, like what Dr. Darshan had mentioned, this work suggests that perhaps different sample that collected from different places, or also different time or different season, would influence the contents of the metabolites or alkaloids in the plant, and hence the standardization is rather very important. And they also look at the content throughout, like what we similar to what we asked, from young leaf to mature leaf. What is the content of the alkaloids? In this case, mitragynine, as example, is a high content in a mature leaf, while the mitragynine precursor, which is cornea gene, it is high in the young leaf. And they also look at different component, leaf, stipules, and stem, and also roots. The content of the active compound, the 10 active compound or 10 active alkaloids in the plant. Are there different tissue type producing different amount of metabolite? Actually, this question we also asked at the time when we proposing to work on young leaf and mature leaf. And the study found that mitrokinin, for example, is not found in roots. So these data that published in this paper show also leaf and also stem and roots are different tissue producing different amount of content of the active compound, and clearly show that yes, the variation is there if you are using different part from the plant, and also when the leaf is plucked or collected at different age. And we always interested in how Ketum synthesis the monoterpene into alkali and also the mitrokinin. So as I think Zuraidah and Dr. Darshan had mentioned, there are 58 alkaloids that actually reported and also a few metabolites. Just for everyone information, I think apart from alkaloids, other metabolites are very little study also in Ketum. So most of these metabolites or recorded are based on the mature leaf and also commercial product. So the leaf that used also, we didn't know when they actually pluck the leaf. Presumable they collect the mature leaf because it's big, so that it is more weight, more molecular mass, and can sell with more price, right? So we are, if this slide show the general pathway in the beginning, what precursor is needed in order to generate mitrokinin and also other similar intermediate also derivative look for the mitrokinin. So titamine and cycloganin is important, and to produce a streptocytin, and which is a precursor for the rest of the product after. Either is indole or oxindole alkaloids that found in Ketum. So the study has been done also, as we know just now from Dr. Zuraidah and also Dr. Darshan, the function of mitrokinin, and the docking research also has been done, reported in 2016 by Google Andrew et al. On how the molecule bind to the human immune receptor, for example. And in that same paper, it reported that mitrokinin or MG could have a lower risk in respiratory depression, as even though it recruit or bind to the immune opiate receptor, and it recruit the G protein signaling, but not beta-arrestin signaling, which normally give respiratory depressions. So with that, it would have a less respiratory depression and tolerant development. So this is up to 2013 when we understand the upstream pathway, ischemic pathway that involve, and also the monoterpenoid scolytoid pathway that involve to produce a precursor of tryptamine, and also cycloganin, and that important for streptocytin, and many pathway, a long pathway that involve to produce mitrokinin. But recently, and the enzymes that involved in this upstream pathway are also identified, but most of the genes are, as shown in the pathway, is upstream, in the upstream pathway, but very few that actually downstream over here. So recently, I think the works that also Dr. Darshan Singh and that involved with several researchers from CDR in 2021, with the first author Nelson Chia published proposed a new biosynthetic pathway of how the alkaloids or monoterpene interalkaloid produced in Ketum. Nonetheless, many enzymes that listed in the pathway are genes that involved in producing the enzyme for synthesizing this alkaloid in the plant are still not known. And interestingly, in the same works published, oxyndoalkaloid cholinosine were also reported to show a high affinity for the human milk opioid receptor. And for mitrokinin is an indoalkaloid, yeah, or 7-OH. So recently, the very recent 2023, a few months ago, the works from Salah Cornell, they also would like to understand how the mitrokinin actually produced. And you will see over here that mitrokinin having a stereoisomer at the C20 position. So where speciogynin, very similar to mitrokinin, but it's a stereoisomer, is not an agonist for the human mu receptor, but it's a smooth muscle relaxants. So how the plant actually producing or very similar compound, but with a stereoisomer. And if you look at these two structures, there are the stereoisomer differences is over here. It's the C20 position. So they did an intensive study and found the two genes in the Ketum, or by mining the genome or the genes that are available in the transcriptome and genome, NSDCS1 and MSDCS2, and with another enzymes called 1-eno-O-methyltransferase over here, which can convert streptozidine aggregon to two distinguished stereoisomer, which is mitrokinin and also a speciogynin. So this is very important, where we knew now from these works that this report that there are dedicated enzymes very specific to produce mitrokinin in this case, and it could actually convert if mutation imposed. And they also study the mutation or active site that may play a role of, for example, by mutating the gene and could produce high mitrokinin or high speciogynin instead of mitrokinin, for example. So means that it is possible to modify or edit the genome if possible. Then one can then use the plant as a factory to produce alkaloids that we would like to have, right? So are there more to know about the plant? As I mentioned, do we know entire profile of the plant? I think the answer is clear. And also there's no study at the time we would like to, when we want to conduct, there's no systematic study on comparing the young and mature leaf. And just to tell a story, why we begin with a young and mature leaf, because we heard people say a young leaf may not have a kick, or mature leaf have a more effects. So we thought that by studying young and mature leaf, we could see how the plant actually producing the alkaloids compound, active micro compound, with the age of the leaf, associated with the age of the leaf. And And also, like what Dr. Darshan had mentioned, many study in Ketum are focusing on alkaloids only, and also for it's a targeted isolation of alkaloids. So we would like to use an untargeted approach to study this. So this work was work of a PhD for Dr. Luba. He just graduated, and we're studying the young leaf and also the mature leaf. We'd like to share a bit of what we found from the study. And we'd like to thank, of course, Prof. Sharif from CDR here. Without him, this work also cannot be done because we are using sample from also from CDR at that time. And currently, we already, like what Dr. Zuraidah have shared and Dr. Darshan have shared, we also went through the process of getting the approval from KKM in order to able to handle and work with Ketum. So I skip this technical metabolomics analysis slide where overall we detected 86 metabolites features and 63 which is assigned to alkaloids and 23 for other metabolites of the ketone. And as you can see over here that not all the alkaloids, or not all the metabolite is sharing by all young and mature leaf. There are distinguished metabolites that you could only find in young and or in mature leaf. So we also can see by comparing the quantification that in a mature leaf, generally you have higher amount of metabolites compared to young leaf in this case. And we further classified the alkaloids, and we knew in the field that largely reporting the indole or oxindole alkaloids for ketone. But there are other classes of alkaloid that from our analysis. And of course, we need further validation for the works, especially to determine the structure of the metabolites that we have assigned to our mass spec data. And there are few significant alkaloids, 5 most significant alkaloids in comparing young and mature leaf. So most of the alkaloids categorized in various subclasses in Ketum are found to be more abundant in mature leaf. And here show the comparison of metabolites or alkaloids, the differences. And in this case, this is high in young leaf, and while another 3 over here is in mature leaf. And of course, we want to see in comparison young and mature leaf, what's different between Ng and also the 7-OH-Mg, 7-hydroxyl-mitrokinin, right? They actually don't have very much different, 1.24 for mitrokinin, but higher for 7-OH, which is about 3.234 differences the amount. And why we are interesting to compare, because 7-OH-Mg it has been known to have a more potent than mitrokinin. We'd like to see whether the young leaf also producing 7-OH mitrokinin. So from the newly identified secondary metabolites that found in the Ketum, perhaps there are other pharmacological properties of the plant that we yet to be explored. For example, a few listed over here with the function like antidiabetic, antitumor. And the list is actually extant, which is reported in the paper. So this may also extend our view on the potential of the plant, apart from what have been emphasized for the properties of mitrokinin and other alkaloids, even though they are in a small, minute amount, and it would worth to explore as the plant is very rich in producing active or druggable compound. So just to summarize, conclude what we have, the work of presented over here, where more alkaloids actually can be found in the plant, and young and mature leaf would producing exclusive molecule. So this could be age dependent, of course. For information, the leaf actually is from the same trees and from the young and mature leaf of the same tree and collected at the same time. So I think this is a good comparison that the age may have play a role. The age of the leaf may play a role in producing responsible for metabolites production. And I would like to summarize what I have shared over here and how we are going to move forward. And for our works, of course, even though we identify newly identified metabolites or molecule that could be potentials explore in its pharmacological properties, we yet to validate the structure of it, and we know that there are many isomer or stellar isomer in the plant. So the bioactivity experiment is definitely crucial to confirm the activity of those metabolites. And DNA barcoding, as what just now we have discussed, Dr. Darshan had also mentioned, we don't have this, we have yet to characterize or categorize our species in the country. So I think that is important. And standardization, have both panels have mentioned. With knowing all the background I share from papers that have published recently, we know that the diversity of the plant in producing the amount of mitrokinin and also the repertoire of the alkaloids. I think it is very important to have a standardization and to identify actually which plant to use, and even for the research study, not only for the use. And the genes that identify and through the genome and transcriptome now can be further explored to understand how the alkaloids biosynthesis pathway works in the plant, and also can be used in the synthetic biology, as we know that yeast or other eukaryotic organism microbe has been used for producing active or drug compound. So the understanding of how the mitrokinin by itself bind to the receptor also not yet been reviewed, even though docking show such a binding of the active compound mitrokinin to the receptor, but yeah, yet to be explored or validated. And it is important to support the function of the mitrokinin in this sense. And the genome editing could be done by showing by the paper that with alcohol dehydrogenase can determine which isomer of the mitrokinin can be produced. I think it is important information over here that we need to explore. And of course, all these will come with the challenges. I think we can discuss after. Really thanks to everyone that involved in the projects for what I have shown, especially Luba and co-researcher over here, Prof. Ayn, Dr. Go, and Dr. Isaac, Dr. Hassan, Puan Salah, Encik Fauzi, and Dr. Arif, and also especially thanks to Prof. Sharif and also Shabina, and Darshan is here at the time when we visit CDR. Thank you. Thank you everyone for listening.
Okay, thank you, Dr. Leong, for the insightful work on Ketum, especially at INBIOSIS. So thank you so much to all the three panelists for sharing with us your knowledge, experience, and opinion about Ketum research in Malaysia. So for now, I would like to open the floor for question and answer session. So I think I'll start the ball rolling. Okay. So as we know that as Dr. Darshan mentioned about that to be a pioneer in a Ketum research, right? For we know that in order for us to register for the product, okay, we need to go through the NPRA regulations, okay. And said to say that metragynine is a substance that not allowed or prohibited in the product to be registered under the NPRA. So metragynine is a main compound in Ketum actually. So how we should go about this? If you want to register a product based on Ketum, so how we can do? Okay, maybe open the question to the panel. Maybe Dr. Darshan, if you have insight on this.
This is a very, very complex question. Okay, but it's very important. I don't know whether I would be able to reply correctly or not. But again, like I was telling you, now you can actually register Ketum product containing mitragynine. Now, mitragynine is regulated under the Poisons Act, right? Like I was telling you, everyone is assuming that mitragynine is one of the most hazardous or one of the most dangerous drug or class of drug compared to other illicit drugs. So I think we have to invalidate this statement by conducting some kind of research, maybe local studies, because internationally people have actually done research to show that mitragynine is not as dangerous as morphine, right? So it's no point harping on this issue, right? So maybe local policymakers are not convinced enough. They always believe that Ketum should be regulated. So like I was telling you, what we have to do now is we have to find ways to conduct some kind of research to educate people and get Ketum or mitragynine. You don't need to, you can remove mitragynine from the Poisons Act, but again, like I was telling you, it can be abused as a narcotic. So again, you have to still regulate mitragynine, but like I was telling you, some kind of leeway should be given. Even morphine is regulated under the International Drug Conventions, but people are still using it in clinical settings to treat pain, correct? So it doesn't mean that whatever that has approved NPA's approval, you can actually buy like vaping punya product. Tidak. It still have to be regulated, correct or not? So this is another tricky or complex question. I have no proper answer. Probably I have to think very hard based on the current context, and then I would be able to provide a conclusive statement. But at present, like I was telling you, please do not take or think that mitragynine is so dangerous as morphine, because when you say mitragynine is more dangerous in which perspective, dangerous in which angle? I have not seen people being rushed into emergency room with mitragynine overdose. Tak berlaku, right? They have a huge problem in America to do with fentanyl overdose. Though though Ketum is also widely used, but I've not seen people dying of mitragynine overdose in America. Even there are cases, but again, we have to remember patients, the deceased is found to have multiple drugs in the system. So I don't know how, but again, like I'm telling you, Ketum is not as dangerous as other illicit drugs. And if we are serious to develop the Ketum industry and then, come up with new novel treatment to treat opioid use disorder and pain, then we have to do something lah. Seriously, we cannot be, saying mitragynine is under the Poisons Act. I cannot register my product. And this is what the former health minister did mention, that they are serious and they want to legalize CBD, for example, and then gradually move into legalizing Ketum also for medical treatment. So maybe the government is receptive to the idea, but again, like I'm telling you, it is a very complex thing. I think so. This can only be answered by policymakers in the country, lah.
Okay, thank you, Dr. Darshan, for the lengthy explanation on that. Maybe other panel, if you have any opinion on this issue.
Maybe I would like to add more to what Dr. Darshan has said. It is true. It is up to how the policymaker looks at the issue. If they always look at negatively, so we will be at this stage for a very long time. So we are not moving. We are not going to see any tangible product out of our own native plant. I think we should take advantage of what we have, because we could see how Indonesia gained a lot of income by selling ketum leaves to the United States, and they allow the farmers to sell it. But our farmers has to sell it illegally to the Thailand part in order to generate the income. So it's not going to solve any problems. I mean, like, the social issues will always be there. When I joined like a certain workshop in order to for them to discuss to legalize ketum and also cannabis, we all has like mixed reaction, especially between the researchers and also the enforcement, especially with the AADK or also the KDN. So they look at that they need to put on, I mean, like, maybe they see that they need to put more resources such as time, money, I mean, like, financial, in order for the enforcement to be stricter, so that the social issue could be reduced if things get legalized. So that is my take on this.
Okay, so we move to the, thank you, Dr. Zuraidah. So we move to the question from the floor. Okay, there's a question from Aisha. Since the 80s, there are account of hyperpigmentation in the users of ketum, and recently reports have been emerged from the US regarding such incidents. Can you elaborate what contributed to this phenomenon?
All right, so maybe I will try to briefly explain about this very rare occurrences of hyperpigmentation. When I first started conducting ketum research back in 2007, I get to see ketum users, especially those who chew kratom leaf or ketum leaf. They have a hyperpigmentation, a darker cheek, right? This is very common or prevalent among people who chew ketum. And then gradually, I also get to see people consuming ketum excessively, developing some kind of black spots on their cheek, but it is not apparent on the entire body. So it only happened on the face region, which is on the cheek. But then not much attention was given to that, because I was assuming because they are consuming ketum, they work under the blazing sun without a cap, so there's a high possibility of developing black spots or darker spots on the face region. But again, it surprises me because it only happened around the cheek region. But again, like I'm telling you, I have not done any specific study to determine why, because even I come across chronic users of ketum, some of them, or most of them are not exhibiting the hyperpigmentation issue on their cheek region. So therefore, not much importance is given, but it is good. Probably we could actually investigate this with clinicians lah. As a social scientist, my skills are limited. But again, like I'm telling you, it is not something which is serious. It's not linked to cancer. And then the dark spot, right? It is not that obvious, it's not that obvious. Like sometimes if you get to see someone who sleeps a lot, they develop hyperpigmentation or darker spot on the cheek region. So it is not as dark as people who sleep excessively. So it is good, Aisha. I will try to study that with my colleagues who are medical doctors to see, try to determine what are the contributing factors to the hyperpigmentation issue lah. Yeah, but for time being, I think so we don't need to worry much, because I've not seen Ketum users saying that, you know, I have developed cancer of the skin. So it's nothing worrying. So but it is good lah that I could do something after this. Yeah.
Okay, thank you, Dr. Darshan. Okay, so there's one comment, maybe the one that can have opinion from the panels. Okay, Ketum perlu dikawal selia oleh kerajaan seperti produk tembakau, alkohol, vape dan lain-lain. Seriously, Malaysia perlu framework on this industry. So maybe comment from the panel.
Like I did mention earlier, I think we still need a proper intervention or a proper kita kata policy lah, because now because sooner or later the government is going to decriminalize narcotics in the country. Berarti people who are dependent on drugs will be allowed to possess certain amount of drugs for personal consumption. We are moving towards decriminalization. So most likely when drugs are decriminalized, Ketum pun akan decriminalizekan. Betul kan? So for example, in Thailand, Ketum is legal, right? But the government has proper framework, has proper interventions to prevent people from abusing, misusing Ketum. You can sell Ketum leaf in the community, but you cannot openly sell Ketum leaf or process. I mean, you can sell, but not in public area. And furthermore, you're not supposed to process Ketum leaf and turn it into a decoction, right? So, but if you want to have a product, then you go through another stage, because you have to register your product, right? And then you can actually commercialize it. Maybe it is a Ketum drink, yeah, but you need to get the relevant authorities approval. So now, like I'm telling you just now, if we don't have a proper framework, then there's no end to the Ketum abuse problem, because now even Thailand dah legalize Ketum. The organized crime syndicates in Thailand, they're not interested to purchase Ketum leaf from Malaysia anymore already. You go out and you talk to Ketum planters, they are telling you no one is actually buying. Who is buying are the local traders, right? So I think so we still need to have a proper intervention, slightly rigid, because we don't want people to start selling Ketum leaf in pasar malam lah. Correct or not? So you need a proper framework. So that is why, like I was telling you, you need to have a dedicated dedicated council or committee to sit and decide. Because end of the day, like Siti was saying, you bring in different enforcement agencies. The National Anti-Drug Agency is saying one thing, the Royal Malaysia Police is saying one thing, researchers is saying one thing. And end of the day, because of the ambivalent views, we cannot make a decisive decision. We come back to the same, we come back to the same spot again, always arguing for the last 23 years, 20 over years since Ketum was regulated under the Poisons Act until today. Kita cannot make a decision. What is so difficult, right? So, but again, we don't need to follow Thailand and legalize Ketum and allow people to misuse it. But again, like as I'm telling you, we have to also regulate the Ketum planters lah. Because if you were to decriminalize ataupun legalize Ketum contoh, then everybody wants to go into the Ketum business. So that is why a proper framework is needed, but the framework has to be formulated to suit the current context, right? Because kita just tak boleh copy what Thailand has done, because it might not suit us. It might lead to serious problem, unanticipated problem. So we have to design it properly. Yeah.
Okay, thank you, Dr. Darshan. I just back from Thailand actually, and then we can found that many store can selling the cannabis. Okay, cannabis. They have a cannabis tea, cannabis coffee, and also many drugs on the shelf on the cannabis as well as the Ketum. So you can sell Ketum like how milligram or microgram of metragynine content in that powder or liquid product of Ketum. So it's everywhere in Thailand. So it's legalized there, yeah. But as I agree with Dr. Darshan that we cannot just follow Thailand like that straight away. We need to have some framework, some process, some procedure to make sure that there is no other the post issue of using Ketum in Malaysia as well. Okay, so there's one question. Okay, good morning, Dr. Darshan. I want to ask your opinion. Do you think Ketum can at least minimize drug problem in Malaysia?
Of course, Ken, it is helping. Ketum is being used from a harm reduction perspective. Sama seperti methadone, right? So a lot of my patients were using illicit drugs, right? They are actually using Ketum for a beneficial reason. Ketum promotes self-abstinence, right? So if I'm dependent on drugs and I don't want to go into a treatment facility, who wants to go into a forced treatment facility, right? So what they do is because they don't want to be associated with treatment, or they don't want to come into treatment because there's so many treatment barriers out there. So what people who do, I mean, people who use drugs, what they do is when they are sick and tired of using illicit drugs, they choose Ketum, right? So they use Ketum to self-treat their substance use disorder, and Ketum do help them, to gradually abstain from illicit drug use. So for me as a researcher, I have done studies with people who use drugs, and I believe that Ketum is being used from a harm reduction perspective to minimize some of the what they call that some of the health issues, social issues, economic issues that are being tied with illicit drug use, right? So for me, I've seen Ketum have in fact significantly helped people who use drugs in the country. So it helps lah to address the drug abuse problem. Yeah.
Okay, okay, many question now. If it okay, from Dr. Hamza, in your opinion, does metraganin or other alkaloid that contain in Ketum may have potential to be used in the treatment for the neurodegenerative diseases, perhaps in the future when more findings have been scientifically proven? Maybe Dr. Leong, you can answer this.
Yeah, thank you. Thank you, Pengurusi. Yes, I think because I see, as I presented just now, we see the plant, the Ketum plant, it is having a variety of active compound. And I think this is a very rich plant in a way that it's at a pot of perhaps drugs that it could be explored and used. And people have used it for many years and show different effects. And what we don't have is that from the scientific point of view, which molecule and how much of the molecule and use or apply can tackle what kind of problem. So the detailed interaction or molecular interaction of when after we consume any molecule to the body, it is not there. So what I can see is, as I shown just now, have not only that 58 alkaloids that already people presented or reported, we also see there are more perhaps. And we can see the pathway also can perhaps produce more. And what behind this is that with the alkaloids or MG, which is has been shown to have effects in male receptor or other neuron related receptor, I think there's a potential there, but we really need to put focus and efforts to discover based on scientific methods, and to provide the evidence on how to use certain compound, because we are now only focusing on mitrakainin or even 7-OH, right? Mitrakainin. And there are molecules that just now I showed in different cultivar or different variety that actually have minimum or low amount of Mg, but other dominant by other alkaloids. So those are also yet to be investigated. I think there's a yes potential there.
Okay, Dr. Leong, thank you. Okay, there's a question from Dr. Ili. Is there effort in organizing team for the intervention as well as advising on the proper framework in Malaysia for plant like ketum or cannabis to be legalized in Malaysia? Maybe Dr. Siti Zuraidah, if you can answer this.
I maybe might just
Dr. Chyan. Okay, sure.
sorry, intervene. Now the thing, it's like me.
Sure.
I've been doing Ketum research in the country.
Mm.
Nobody have ever invited me to come and sit and share my ideas, right? So this is the sad reality in the country. Kita yang buat kerja dengan pengguna ketum, duduk dekat longkang dan sebagainya, tak pernah dijemput. And and they make all kind of decisions. So this is so sickening. Now again, yeah, there is a possibility kalau ketum researchers can engage with the relevant authorities and policy makers in the country. I think overnight we can come up with a proper framework and find a permanent, long-lasting solution to the ketum controversy in the country. Now, coming back to your another question, you did ask about do ketum have the sign effect same as the drug? Okay, let me just share with you this. I've not seen people who use ketum developing mental health problems. I've seen people who use crystal methamphetamine with serious mental health problems. When they use ketum, right, they are more calmer. There is no violent outbreak in them, right? So what does this indicate? Ketum is actually helping them therapeutically, to just stabilize their mood imbalances. So this is what Siti is doing, that she wants to look into depression, because depression is one of the primary or principal withdrawal effect in methamphetamine users, right? So you just imagine, I sometimes do receive call, mothers used to tell me, anak saya guna ketum okey ke tak doktor? I said, bagus kalau dia minum ketum sebab dia tak akan jadi ganas, right? So there is something here, because this is live experiences of crystal meth users yang tak ada masalah gangguan mental ataupun psychosis or whatsoever. So I think this is where research has to come in and explore this area lah. Betul kan? So now again, like I'm telling you, saya tak pernah tengok orang yang guna ketum develop serious mental health problems. Tak pernah berlaku. Right? Yeah.
Dr. Darshan, how about the euphoria effect? There is euphoria effect.
Okay. Let me just tell you very simple lah. Sebenarnya kalau kita guna, if we were to consume pure ketum juice, pure ketum juice bererti bukan cakroy. Kalau you consume pure ketum juice, of course you will experience very mild euphoria, right? But why macam kita kata mengapa cakroy become so famous? Because they like to add cough syrup. The moment you add cough syrup with your ketum juice, of course lah, the kick is better. The euphoria or the euphoric effect is much more stronger. And these people who use ketum, because when they know that the cough syrup, when you combine with ketum, it's not giving you 100% euphoria ataupun euphoric effect. The people who are trading ketum, they also sell Aramine 5. So bila kita makan Aramine 5 and on top of that minum cakroy ataupun ketum jus yang telah dicampur dengan ubat batuk, of course lah dia punya steam is so high. But again, kalau you consume pure ketum jus, benda ini tak akan berlaku. It only induces very mild euphoria. And dia punya effect is just very short acting sahaja, and then it turns into a stimulant, right? So but macam saya katalah, bila dia minum ketum, tak ada kick, tak sedap, tak ada high. So kalau saya nak membazir duit, nak minum ketum buat apa? Tak ada kick. So what they do is campurlah dengan ubat batuk. Semua pot ketum, they are selling ubat batuk. Dia ada cocktail, right? Yeah, so this is the problem. So bila media buat laporan, dia kata ketum ni boleh menyebabkan halusinasi. Sebenarnya ketum bukan ketum pesi, sebab ketum yang telah dicampur. So they don't really understand. But again, like I'm telling you, bagi seorang penagih, for a drug user, macam heroin user, okay, ataupun kalau kita kata poly drug user lah, if they want to abstain from drugs kan, kalau you pergi bagi dia ketum, oh memang tak cukup untuk dia, because this is not a therapeutic formula to treat opioid use disorder. This is ketum decoction sendiri masak, and the efficacy could be just 30% saja. Correct or not? So that is why it is so important for us to develop a therapeutic formula to treat heroin addiction or pain, so that patients receive 100% efficacy. So bila kita minum ketum decoction, kita pun tak tahu whether they receive 20% punya therapeutic value ke, 30% ke, 40% ke. Of course, ketum help to suppress pain in heroin users. Cuma yang mereka rindu adalah high. Betul kan? So takkan nak minum 3 liter ketum tak high-high. So what they do, they mix lah cough syrup, murah saja, bukan mahal. Can go and buy over the counter. Beli baru high. So you see, this is what is actually happening. They are trying to adulterate the ketum juice. So that is why you need to have proper interventions to actually address all this, right?
Okay, thank you, Dr. Darshan. Okay, so the next question, how can we help the local ketum farmers, for example, untuk dapatkan license for their plantation selain daripada didenda oleh certain agensi penguatkuasa? I can answer the question. How to help the ketum farmer?
Siti wants to answer.
Yeah, okay. The thing is, if we look at the Poisons Act, technically dia tak cakap that we cannot plant the ketum. That's the thing. It is it is wrong when we pluck leaves and sell it. So planting is not something. It's not saying that it's illegal in Malaysia. That's the thing. So I'm not sure how to, unless like the government or the enforcement sees differently to make a license out of it, then we can actually, I mean, the farmers can actually pluck the leaves to sell it to the customers that need it. Because the thing is, if they plant a lot of trees, but they cannot pluck it, so there's no point as well.
Now, I have an idea, but I don't know whether this is feasible or not, and whether people are going to support or throw brickbats at me. I believe kalau mereka yang ada ladang-ladang ketum, those who are ready, those who possess ataupun own big ladang-ladang ketum, or whatever small ladang ketum dengan 100 pokok ataupun 200 pokok, I think so this ladang should be regulated. They are supposed to be strictly regulated, berarti that we tell them that you're not supposed to sell your raw material to any illegal trader. You have to sell to a government entity, or you sell to someone who is interested to buy from you the raw material. And you should prevent and stop new people from coming in or venturing into the ketum business. Sebab there are hundreds of plantations. So you don't want to convert the entire northern region state into a ketum plantation. So one day you will see nobody is going to plant rice also. Nobody is going to plant padi also, because you don't make profit. It's not a profitable business. Correct or not? So I think so when you come up with a framework, you have to do it very fast. You have to also learn to prevent people from applying licenses to set up their own plantations. So those yang ada ladang, it is their luck, because they've been doing this business for very, very long years. So we regulate, but we tell them strictly, you're not supposed to, cari makan lah. You sell to the government and then you also sell it illegally. You're not supposed to be doing all this. If you were to do that, if you are being caught red-handed, we will revoke awak punya license. right? And then very important is these people, macam Dr. Siti dengan Dr. Leong pun ada kata, the leaves are contaminated with heavy metals. So you need to come up with a proper planting guideline and tell to these people to treat dia punya soil ke whatever, and make sure they comply ataupun follow proper planting punya practices, and they will know exactly when is the right time to harvest. Maybe harvesting boleh buat 5 kali setahun. So we have no idea now. Sebab sekarang, kalau permintaan ketum tinggi, if the demand for ketum leaf is high, sometimes they just plug whatever which is 3 weeks old pun dia letak dalam daun 5 minggu, and then they tolak kat you, and then you drink and you masak and you drink. And so this is what is actually happening in the pot. Every day, ketum users are telling me, kesan ketum yang mereka alami itu berbeza. Ada hari mereka hanya beli 1 paket, ada hari mereka terpaksa beli 2 paket, ada hari mereka terpaksa beli 3 paket. You see, they are also experiencing the difficulty. It is not us here, having what they call that variations in the nitrogenine content. So again, like I'm telling you, whatever life experiences that are being shared by people who use drugs or people who use ketum for a medical reason, we have no idea whether they are obtaining 100% efficacy ataupun 5% punya efficacy ke 20%. We have no idea because we have not even come up with a formulated ataupun developed or standardized formula. So it's very important for us to move towards this direction, and then kita buat clinical trial to prove. So we have to start from somewhere lah. It's no point arguing it. We have been arguing for the last 20 years already, and there's no permanent solution to it.
Maybe we still arguing until another 20 years.
Correct.
Okay, there's a question, Dalila, do ketum have the side effect same as a drug to the mental health state?
Like I did mention earlier to Dalila's question, I think like I did tell Dalila, I've been working with people who use ketum in the community. Saya tak pernah tengok pengguna ketum develop psychosis ke, pengguna ketum jadi mental ke, ganas ke, depressed. Of course, kalau a chronic user who have been abusing ketum for prolonged periods, chronically every day, when he abstain abruptly, there is a possibility of that patient developing some depression. Of course, dia akan experience a bit of depression. So that is what I did mention in my challenges, that it's very important, because in the future, if ketum is decriminalized, kita punya primary health care punya facility will be frequented by people with ketum use disorder. So we need this treatment guidelines. Saja kata Dr. Darshan pengguna ketum, have developed ketum dependence, happen to go to a primary care to seek some kind of therapy because saya ada depression because I've abstained from ketum. So there is a treatment guideline advising the medical provider what to do. I could be treated, right? So that is why we have to look into all this if we were to move towards legalizing ketum or decriminalizing ketum.
Maybe I would like to add a bit.
Okay,
Maybe
sure,
I'd like to
Dr.
add a bit. Yeah. I think towards all these open questions, of course, thank you, Dr. Darshan, for sharing the experience when with the ketum consumer. But I think another angle is that we really need scientific evidence to support those side effects. If it's come from where actually is affected by the molecule that you consume. Because as we see, if one taking the ketum extracts, you have perhaps hundreds of active compound or molecule that you're taking in, and where this compound actually works. And I think that's have to be really studied clearly and intensively studied, and not only focusing on Mg. Of course, mitragynine is most dominant molecule in general, but not all. We have seen, right? Different cultivar or maybe different time we harvest may be different. So all these need to be really well studied, not hearsay whether we have seen or we have, but we need scientific evidence, and we really need more funding perhaps to support these works, as we see that if the plant is really potential to develop various type of perhaps drugs, right? So I think that we need to have act as such to address these questions.
Okay, Dr.
I
Yan,
think
thank you.
I would suggest INBIOSIS should take up the lead, bring up this matter with the relevant authority and get the right funding. In fact, you could maybe work together with us and become, Ketum Research Institute and lead the project, because like I'm telling you, the Ketum research is not streamlined in the country. So since you have organized this webinar, why not lead the project? Everybody wants to become leaders for their own personal benefit. So why not get everyone collectively work as, a big family and get things done quickly?
Thank you, Dr. Darshan. Okay, just to let you know that last year, if I'm not mistaken, we have a meeting in the UKM. So inviting all the people that are working on Ketum as well, different faculty, law faculty, pharmacies, medical faculty, economy faculty, to discuss about this and how to bring the policy of Ketum to the cabinet. Okay, but that time is under the KJ, okay, Khairul Jamaluddin. Okay, but since we have changed the government, so I think INBIOSIS maybe need to do it again, to bring this the matter on the policy to the cabinet again. I will talk to the top management of UKM, see how we can do about this. Yeah, thank you for that, highlighting this. Yeah, we should do things maybe with the different government also, but still we need to do basically based on the scientific and also from the social science perspective. Okay, there's a question from the FB Live, Arifin. In order to make Ketum as a legal consumer product, comprehensive regulation is needed. Is there any regulation formulated or suggested for Malaysia? Maybe Dr. Siti can answer, because you are the technical committee for the Ketum industry, pembangunan industri Ketum, right? Okay.
Okay. So for that question, like I mentioned in one of my slides, the thing is under the RDG, the drug registration document, it has stated that any product that comes from this plant, that Metragana spiosa, is totally prohibited from being developed as a product. So that is one thing. But in optimism, if this being legalized, so MPRA also has the regulations on how to develop botanical drugs. So it can follow that regulations if Ketum itself become legal in Malaysia. Yeah.
Okay, thank you, Dr. Siti. Okay, so we move to the next question. Okay, there's a comment. Not the Ketum juice per se is mostly due to the Ketum cocktail. Okay, so you already know about that. Okay, there's opinion from the FB Live, Anna. Okay, maybe government should build an institute like palm oil or rubber, which is specialized in handling Ketum. Then Ketum supplier is registered and monitored their upstream and downstream supply chain. Like Dr. said, it's required many enforcement. Okay, I think Dr. Darshan already touch on this part, right? Okay, maybe if any other opinion on that?
No, I think so. It's very important for us to be governed by a proper body. So a Ketum Research Institute has to be urgently set up to look into this. The plight of Ketum researchers, the plight of Ketum planters, the plight of Ketum users, right? So when you make a policy, you have to make sure that you invite all this different population of people to sit on the table to draft an intervention. So you cannot always exclude the planters, the users, and hope and assume that, whatever laws that you come up with will actually cater to their needs. No, we will always be in conflict. We will always be in dispute. So that is not doing things rightly. So I personally believe it is about time We call the shots. Research has been done to show that Ketum is not as harmful, or it is not being used as a primary drug in the country. If Ketum is being used as a primary drug in the country, we have won the drug battle. Kita tak ada orang yang akan guna heroin dan crystal meth. There's no trafficking of marijuana dan sebagainya. And the sad part is this morning, if you read the Star paper, for the last 3 days, Royal Malaysia Police have conducted crackdowns, okay, on drug dens. They seize two most drugs widely used, which is marijuana and Ketum. Again, why go and attack Ketum pods? You see, so basically, you cannot find anyone using drugs and you attack people who use Ketum, because as I'm telling you, Ketum is being used by people who are using drugs to come out from substance abuse. So I think so we should take this matter seriously, come up with a proper institute to also come up with a proper a proper law or a proper policy to look into all these nitty-gritty issues lah to do with Ketum use in the country. Yeah.
Okay, there's one question from Ashraf. Kratom suitable for animal consumption? Is it? We never try on animal, right? Okay. Maybe if you have any experience on that.
Okay, coming back to Kratom, right? When I used to go into the kampung, into the community, I always see mereka yang rare kambing, rare lembu, right? So they always, or buffalo, they all normally akan bagi Ketum leaf, tetapi bukan frequently lah. Ada pada masa-masa yang tertentu, mungkin lembu sakit dan sebagainya. So this is what they told me. They said kalau kita bagi daun ketum, lembu mungkin dapat menghasilkan susu yang banyak. Contoh ini yang mereka bagi tahu lah. Tetapi scientifically we don't know anything. Mungkin lembu yang guna ketum ataupun kerbau yang guna ketum rajin bekerja seperti manusia. Kalau mereka yang guna ketum rajin kerja tengah panas, it's not easy to go and stand outside. Sometimes when I interview ketum users under the sweltering sun, blazing sunlight, 10-15 minit kita dah peluh, dah panas dan mereka can withstand the heat. You see? So yeah, so of course it is being used in animals juga, but again macam it's a folk law lah. So they use for their own particular reasons. But again, I think so we should not give more importance on all this. We should just move towards developing ketum to treat pain and opioid use disorder and probably mental health problems in the near future. Yeah.
Okay, thank you for the answer. Okay, there's one opinion from the Facebook Live, Hunger Love Official. Really looking forward for ketum being legal in Malaysia, since there's a lot of good side compared to the bad side of its usage. Yeah, I think everyone of us are looking forward for that. Okay, I think there's no more question. Okay, yeah, we are reaching 11:50. Okay. So I think that's all from our webinar today. Okay. So we have come to the end of the session. Okay, I would like to thank all the honourable speakers for their willingness to share with us knowledge and experience on the today hot topic. Okay, ketum research in Malaysia. So I'm sure it has opened up our view and knowledge with the regards to ketum research in Malaysia. I would like to thank all the participants for spending your time with us today. So before we say goodbye, okay, let us immortalize this session by switching on your video. Okay, I hope the technical team are ready to assist me on the photo session. Please switch on your camera. We can capture all the beautiful and handsome faces. Okay, just to let you know that we have about 215 participants as for now from the WebEx and also from the INBIOSIS Facebook Live. Okay, so many participants today here. So thank you for that, for your commitment. Nearly more than 2 hours, yeah. So okay, technical team, please help.
Okay, everyone, can you switch on your camera for photography session? All right, I will count 1, 2, 3. 1, 2, 3. Smile. Okay, another one. 1, 2, 3. Smile. All right. Okay, thank you.
Okay, thank you, Intan. Okay. Okay. We have posted the attendance list at the WebEx chat box. Okay, please be informed that the link will be available for 30 minutes only. Then the link will be closed. Okay, you can register your attendance. Then you can have your certificates. Okay, before I end the session, I would like to invite the speaker, Dr. Siti Zuraidah, Dr. Darshan, and Dr. Leong to join the breakout session with our chairperson, MNPS, Prof. Dr. Nohadiani, and other interested researcher. You can join the breakout session if you like to. Okay, kindly PM if you like to join the session, then we can share with you the link. Okay, so kindly wait for the breakout session invitation by the host. Okay, please stay safe and take a good care of yourself. Have a good weekend, everyone. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh. Okay, for the the breakout session is already open. Okay, hope the panel can join it. Okay, thank you so much, everyone.
Thank you, everyone.
Assalamualaikum, Dr. Cik Putih. Are you there?
Yes, I see.
Okay, you can proceed.
Oh, okay. Hello, everyone. So, I'm Dr. Chipite from UITM. So, I believe the audience are still here.
Yes, many of
Okay.
them are still here.
So, I would like to take this opportunity to thank the organizing committee for allowing us, the ICMP um, organizers, to promote our conference this year. So, as you may know, the International Conference on Natural Products 2023 will be hosted by University Technology Mara. The conference will be held at Alof Kuala Lumpur Central. So, you can find more information about our conference by our website, ICMP 2023 .uitm.edu.my. I'm going to paste the link at the chat box. Okay. So this year's conference will be organized for 3 days, and the last day will include the parallel workshops, which is inclusive of the registration fee. So you can choose 3 parallel workshops on NMR, LCMS, or the development of natural products. You can also join the one of the workshops organized by the Institute of Medical Research, Ministry of Health. Okay, so this year we are going to have 5 plenary keynote speakers, and also the speakers of the Jack Cadnan lectures, which is the traditional lecture in the ICMP. So please register now. We are expecting many people from various backgrounds, from the fundamental work up to the regulators. So join now. You can scan the QR code or you can click the link that I've posted in the chat box. Thank you.
All right, thank you, Dr. Cik Putih.